Hello, everyone. Thank you for joining us. My name is Vlad Kovalenko. I'm a master's student in Diana Six's lab at the University of Montana. I'm co-organizing this webinar with Teo Rautu, a master's student in Danielle Ulrich's lab at Montana State University, as well as Laurel Sindewall, a PhD candidate in Diana Tombak's Forest Ecology Lab at CU Denver. If you missed any of our previous installments, you can find recordings of those talks on our YouTube channel, and I will post that link in the chat. And before we get to Dr. Sniezko's talk, we have David Neal coming on to say a few things. Dr. Neal is a distinguished professor emeritus at the Department of Plant Sciences at the University of California, Davis. He's been on the faculty there since 2005 and recognized as a distinguished professor since 2017. His contributions have included Director of Forest Biology and Research Center, Chair of the Bioenergy Research Center, Chair of the Genetics Graduate Group, and Chair of the Agriculture Plant Biology Section. Prior to UC Davis, he was a plant molecular geneticist with the Institute of Forest Genetics at the USDA Forest Service. He's actively involved in many synergistic activities which complement his academic contributions. He's also the chair of the White Bark Pine Ecosystem Foundation Genomics Committee and is pursuing the genetic sequencing of white bark pine and bristlecone pine. He's published widely in genomic research for a variety of conifers. And with that, I'll turn it over to you.
Wow. Okay. Thanks, Vlad. That was a lot more than I thought might be said. But anyway, yeah, I am a retired Forest Service and UC Davis scientist. I'm coming to you today from Wolfboro, New Hampshire, where I live now. But I'm very much involved with the White Bark Pine Ecosystem Foundation, serving as their director. But prior to that, even while I was still at UC Davis, I began the project to sequence the white bark pine genome. And I've been asked just to make a couple of comments about the status of that. I'll begin by saying why sequence the genome of white bark pine. For some people, that's probably quite obvious, but it was really to develop the end goal is to develop modern technologies that can be used to rapidly identify trees that may bear genetic resistance to white bark pine blister rust, and furthermore, identify trees that would be adapted to present and future climates. All this type of work we've done in other conifer systems, notably sugar pine. And for us, it begins by sequencing the genome. Once that's done, you now have a parts list of all the individual genes in the genome, and then you can begin the studies to identify what genes do what. In our case, of course, identifying the genes for resistance to blister rust and also adaptation. Those are the primary focuses. So we began a couple of years ago. Well, let me step back. We've sequenced a number of conifer genomes, by the way, that are very large, 10 times the size of the human genome. So I had to wait for technology to catch up and willingness for funding agencies to fund that work. But we've been successful at sequencing a number of conifer genomes, log pine, Doug fir, coast redwood, giant sequoia. And sugar pine was one of them. And the goal of sugar pine was very much the same as white bark pine, was trying to identify genes for resistance to white bark pine blister rust. Some of you, I think, on this webinar would know that there are two forms of resistance in sugar pine, the so-called major gene, Mendelian and inherited resistance, and then quantitative determinants of resistance. And we were successful in identifying individual genes for both of those things. The single dominant gene, we think we have that, or certainly we have genetic markers that are very closely linked to it, that can now be used in a practical manner. Region 5 is using it for selecting trees for resistance. And then going on to quantitative determinants of resistance. So that's the goal in white bark pine, to find such things. Richard would tell me that to date, there's no evidence for the major gene in white bark pine, but there's certainly quantitative resistance. That's what his whole program and seminar will be about here in just a minute. So we'll use basically the same discovery path to try to identify those things in white bark pine. That it's another tool in the toolbox to assist the type of work that Richard Sniezko and others do. So I'll wrap it up here. We started sequencing the genome just about 2 years ago. Well, it's finished. It's sequencing a genome has sort of 3 major steps: sequence it, assemble it, and annotate it. Sequencing is done, assembly is done, and the annotation is just about done. We're starting to write the paper. And those of you who are interested in having that resource for various things, we hope to be able to release that into the public domain pre-publication sometime this spring. So the step 1, phase 1, the genome will be sequenced, and then we'll funding provided anyway. We'll move on to then trying to discover the individual genes involved in resistance and adaptation. That's actually probably even a bigger project, but it'll take a couple more years to complete that work if we can identify funding for it. So hopefully that's enough for people. And I'll just say that Richard's been involved in all this stuff that I just talked about, and been a very productive collaboration relationship with he and his group at Dorena. The white bark pine tree that we sequenced the genome, Richard went out and got for us through his collaborators anyway. And we'll be working with him, I hope, in those future steps to identify genes involved in resistance. So I think I'll stop there, Vlad, and let Richard begin his seminar.
Sounds good. Thanks, Dr. Neal. It's my pleasure to introduce to you today's speaker, Dr. Richard Sniezko. Dr. Sniezko has been a center geneticist at the USDA Forest Service's Dorena Genetic Resource Center in Oregon since 1991. His current work focuses on development of populations of trees with genetic resistance to non-native forest tree diseases and pests for restoration and reforestation, as well as genetic variation and gene conservation. He has worked extensively with white pine blister rust, as well as Port Orford cedar root disease and koa wilt. He has organized and co-organized 11 international conferences and workshops on 5-needle pine genetics, breeding, and genetic resistance since 2001. Some additional recent activities include providing guidance on the project to develop resistance to rapid ohia death in Hawaii, some genetic and gene conservation related projects with Western species of ash, and setting up a new Sentinel project to help evaluate potential future pathogen and pest threats to U.S. tree species. As always, if you have any questions following Dr. Sniezko's talk, we encourage you to virtually raise your hand so that we can enable you to unmute yourself and start a dialogue. If you're unable to unmute yourself, we can put your questions in the chat and I will read them out loud. And with that, I will turn it over to Dr. Sniezko.
Hello, everybody. Just trying to get the presentation up here. So give me just 1 minute. Okay, hopefully you can see the presentation. It's on my screen. Hopefully it's on yours as well too. Thank you for the opportunity to present here today. I'm going to go through resistance, a little bit what it is, the types of resistance. I do thank Dave for giving the information on the genome of white bark pine, one of the 9 species of 5-needle pines in the U.S. And Dave gave a lot of background on what that can be used for and how genomics and genomic resources might be able to tie with what I'm going to talk today. Because of lack of time, I'm mostly going to talk on the phenotyping and some good news about resistance for some of our 5-needle pine species. So the problem are our 5-needle pine species are highly susceptible to white pine blister rust caused by a non-native invasive pathogen. And in some cases, they can probably kill, in some populations, 99% or even some of the populations. And the solution really is, a lot of things have been tried over time in the early days when Blister came into this country, but the real solution will be find and use genetic resistance and restoration. And in a sense, it's kind of an assistant recovery. You hear of assisted migration. Well, hey, we've got to assist the recovery of the species by finding and increasing the resistance. So there are a range of 5-needle pines, 9 of them in the United States, 4 of them are also present in Canada. Whitebark pine, one of the species, is recently listed as threatened under the Endangered Species Act in the US, and it's listed as endangered in Canada. All 9 species are susceptible to white pine blisterus, but as you'll see, to a somewhat different degree. The white pine blisterus pathogen is not native to the US. It came in about 100 years ago. I'm not going to go into a lot of details on that. And then the pathogen does require an alternate host, and there are several alternate hosts in the US, but the one probably of most importance are the ribes species, occurrence in the gooseberries. This is simply a list of the 9 US species of 5-needle pines. And the photo to the right shows one of the things I'm going to talk about later is field trials and plantings. Well, this is actually a 40-year-old trial of sugar pine, and the same material planted at 6 sites, and probably could be the oldest continuously followed trial of resistance to a non-native pathogen, maybe anywhere in the world, that's over multiple sites. Been followed for 40 years and has some interesting information that we'll be putting together. I mentioned that the white pine blisterus pathogen requires an alternate host, which is ribes, and essentially this is a life cycle. 3 of the spore stages occur in the ribes plant, 2 of them occur in the pine. You have to have the ribes. The spores from there go to the pine. They infect the pine. The pine will produce some spores or sporulate. They will go back to the ribes. So you have the cycle. And as far as we know, you can't have blisterus here without having, you know, both hosts of the pathogen. And the pine is very, very susceptible. So we're looking for resistant. A lot of the ribes are more what we would call tolerant. You can see the leaf on the left is covered with blisterus, but it really doesn't harm, for the most part, the ribes plants itself too. So it's kind of tolerant. We're looking not for tolerance in the pine, we're looking for resistance in the pine. And there are various tools for that one can look at for when a non-native invasive pathogen comes in. And you know, you can look at silviculture, you can look at the microbiome, maybe biocontrol, but what we're really going to focus on, and I think is perhaps the most important one, is genetic resistance. And, you know, traditional selective breeding is a key. It's been used for thousands of years to breed, you know, crop plants and that type of thing, you know, without a knowledge of really genetics. It's a green natural solution to a disease problem in this case. And there are success stories with resistance breeding now. And Jennifer Cook and I kind of review maybe the top ones in a paper in 2017. Photo to the right shows I'll talk just briefly about it a little bit later, a trial at low elevation of whitebark pine that we have planted in 2015. The key to any of this, if you're developing a resistance program, is to bring together certain components. There's a research component, there's a tree improvement component, there's management commitment over time, and then there's a reforestation or restoration. And what I like to say, you know, resistance is a numbers game. So you need some research to start. But, you know, as I tell people, if it stays in research, you're not doing anybody really any good in terms of an applied application. The tree improvement or tree breeding part of it, forest genetics part of it, is a part that is kind of the applied development of the resistance on a scale that can be used for reforestation or restoration. None of this happens in a year or two, you know, not with trees and not with crops. And so you need management commitment over time if you're going to do this and if you're going to do it correctly. And then it comes down to those people who are actually in the field doing the restoration and getting the material out there. So you have to provide seed, it has to get out there. One of the very important component, and that's when you get success, when these four things come together. And all of this is overlaid with a network of partnerships that are essential for success for genetic resistance and restoration programs. There are lots of partners. I'm talking to you from Western Oregon, Cottage Grove. I live in Eugene, and I serve mostly the region in Oregon and Washington, the Pacific Northwest region, but I work with cooperators throughout the range of Blister as well as other pathogens that I work with as well too. So there's kind of two phases of working with a resistance program. The first question is there resistance? And that would seem to be kind of a simple thing to answer, and I don't have time to go into detail here, but it's not quite as simple as it's made out to be. As an example, with Porterford seed or another species I work with a root disease problem, I think 15 or 20 years were spent a long time ago looking to answer that question, and it's actually published out there that no, there is no resistance to Phytophthora lateralis. They were about to give up, had a couple plants that didn't die, tested those again, decided there was resistance. I came into the picture as a kind of a tree breeder soon after that publication was out, and we started an operational program for resistance. We showed that actually there's a fair amount of resistance in Porterford cedar. There's two different types of resistance in Porterford cedar. So that first question is a very important one to answer, and to answer correctly. Then you want to look at the frequency and the geographic distribution of resistance. You want to know what type of resistance there is. You know, Dave Neal talked very briefly and said there's major gene resistance and quantitative resistance. So those are two broad categories. What is the level of resistance? You know, what survival am I going to get if I put this material out there? And hey, it can be from 0 to 100%, depending on the pathosystem and various other dynamics. And then it's complicated because unlike an annual crop plant and row crops that you're putting out for a year at a time and you can replace it at some point, we're putting trees out that, in the case of white bark pine, have to be out there for hundreds of years. So the resistance has to last, even if the pathogen can evolve. And not only it has to last for that tree in the field, but those trees that we're putting out are progenitors of the future generation. So really, we in forestry have to look for resistance that is durable. It has to last forever, is what I tell people. So we take a different approach maybe than some of the folks do in some of the crop breeding efforts. So we want durability of resistance. But there's another term that not as many are familiar with, stability of resistance. And that has to do with, will the genetic resistance you have, is it effective in all environments? And there are more and more papers coming out that show that sometimes just a couple degrees warmer temperature or change in temperature can affect the efficacy of resistance. So certainly in trees, we want to know this. And we have some things going on that will look at this for white pine blister rust and stuff like that too. But we want resistance to be durable because our trees have to be out there for a long time. We want our resistance to be stable over many environments. And with climate changing, even planting in a given environment, that environment will change over the hundreds of years that tree is out there. Will we have stable resistance? So these are some of the concepts. And then we want to know whether it has efficacy on the highest hazard sites. You know, I tell people I want to know what the worst sites are for blister rust because I want to plant some of our materials out there. Phase 2, after you look at phase 1, then you might say, well, hey, do we need to launch into an applied resistance program? Is breeding needed, or can seed from resistant parent trees be used for restoration? Well, Western white pine and sugar pine, you really needed to do some breeding. For white bark pine, you can do a certain amount just collecting from parent trees. We got lucky with white bark pine. Are seed orchards needed? For some species, yes. For other species, it would be helpful, but they may not be needed. How do you protect the resistant trees if you're going to be collecting from the parent trees in the field? And you know, you have to be able to do that. Phase 2, you want to know what the expected survival is, because land managers want to know how many seedlings they need to plant to get X% or X number of trees per hectare out there, you know, to meet a land management objective. So, you know, we need to be able to say what the % survival is expected. Then you need to know who or what group oversees resistant development. And in the US, there are often regional groups. I've served mostly Oregon and Washington, but I do work with programs elsewhere as well too. But there are programs in the interior West, there are programs up in British Columbia and Alberta, et cetera. How to organize the successful applied program. One of the keys I tell people, like when I was over in Hawaii and talking about OHIA and the start of a program there, I said, worst thing you could do is let the program get caught up only in research. Be thinking about the applied development of resistance from day one, and then let research feed into that, not the other way around. And then when will resistant seed become available? And that's very much dependent upon the species and what you find out about resistance. How do you maximize restoration? You know, you're not going to be able to plant every single acre or hectare that was decimated by Blisterus. And when can it begin? Those are some of the questions for phase 2 and beyond. This is an overview. As I mentioned, resistance is a numbers game. Dorena Genetic Resource Center, we are not a research unit. We're actually in the National Forest Service system, and we're an applied development branch. So we look at developing populations of trees, not a single cultivar, with genetic resistance to use for reforestation or restoration. We also, since they're forest trees, we want to maintain genetic diversity because trees face a lot of things, abiotic and biotic, over the hundreds of years out there, and they have to continue to evolve. And we want to make sure we attain adaptability. Many years ago with Porterford cedar, I found a parent tree that had 100% resistance in the greenhouse test, and one of the colleagues I was working with said, great, you solved the problem. You just take that parent tree and you breed it with everything else. Well, it's not quite that simple. You're gonna truncate some of the genetic diversity if you do something like that. Anyway, you can see we do things on a large scale. We may have the world's largest inoculation system chamber for plants. We have, probably hundreds of thousands of plants under test over a period of time. And we can do things on an operational scale. Looking at white pine blisterus, these are some photos of 5 different species showing everything from needle spots, the different types of needle spots, seedlings that get more than one stem symptom or canker on them, seedlings that get bark reactions or hilled cankers, seedlings that drop their needles and maybe have a needle shed trait and whatever too. So there are different things that we look at when we're looking at resistance in 5-needle pines. As Dave mentioned, there are 2 broad categories of resistance: complete resistance, that in white pine blisterus is conditioned by a major gene, which means if you have a parent and its offspring will either give you 50% or up to 100% resistance or survival. Okay, that sounds really good. However, that is only if you don't have a virulent strain of rust that evolves. And the problem with major gene resistance in the 5-needle pines, we have found it so far in western white pine, sugar pine, limber pine, and southwestern white pine. Well, in western white pine and sugar pine, there is a virulent strain of rust. And some colleagues and I, and some colleagues in Canada and I think we have also found a virulent strain of the rust for limber pine for the major gene as well too. So you don't want to rely on only the major gene resistance in 5-needle pines. You can use it, but you want to use it in conjunction with other types unless it were the only type of resistance you have, and then you have to look maybe how to use it. And then partial resistance or quantitative resistance is due to more than one gene. It's thought to be much more durable, and in many systems it is. And in this case, the resistance would vary by among families, and even within a family, different seedlings would have a different type or different levels of resistance there too. The graph on the right is actually from Porterford cedar, but it shows 6 or 7 families. The red line at the top shows 100% mortality very early. The other line show mortality is proceeding over time, but none of them really, I guess one of the remaining 6 or so get up to where the susceptible went eventually. The others, they have tree seedlings die, but they never get to the point of the susceptible. And each of those points on there are different numbers of seedlings that are alive or dead or whatever too. So even within a family, you get variation when you have quantitative resistance. Some of the attributes are traced within quantitative resistance. You may get seedlings that have no stem infections, and there's different maybe traits that lead to that. I'm not going to go in detail. We see seedlings with reduced number of infections, either fewer needle spots or fewer number of stem symptoms. What seems to be happening in many cases is the fungus seems to be slowed down. So we have slow fungus growth, slow canker growth. Once it gets into the stem, it seems to be progressing slower. We have some seedlings that, hey, they get cankers, but they live with the cankers, even though the cankers are active. Some of them have reduced sporulation, so that's reducing things. Some of them have bark reactions. You'll see some photos of that as we go along. And then some of them just have delayed mortality. And you can have all of this within one family that's a good quantitative resistance family. So as opposed to the susceptible family, hey, they all get infected, they all die pretty early. So there's a real contrast. And you can talk about resistance at the level of the individual plant. Is that particular seedling or tree resistant? Okay. And an individual tree or seedling can have complete resistance. It can have the partial resistance, or it can actually have both types of resistance woven into it if you're lucky. Or you can be looking at on a planting stock. Hey, I'm using a seed orchard mix. What is my frequency of resistant trees from that mix? And that can vary from 0 to 100%. Seldom will it be 100%, and it may be low to intermediate. And the type of resistance and level of resistance within the offspring can vary depending, what you're putting out there. What is known is that the frequency in most natural populations of most of the species of five-needle pine in the U.S. are really low. There's some exceptions to that, but generally it's very low. I talked about durability resistance, stability resistance, and there's usability resistance. Durability resistance asks the question, will your resistance hold up if you plant this material over many hectares, over many years, and in areas of high infection? Or will the pathogen evolve to overcome the resistance? So, hey, we want durability resistance. So we want this stuff out there and to test it. How long does it take to test durability resistance? A long time, because things can change, and we don't really understand. And same thing in the crops, for the most part, the basis what makes something durable. Okay, that's why the term that was coined many years ago for a crop was resistance is durable when you plant it over many hectares, over many years, under conditions of high fungal activity. It's durable until it's no longer durable. Okay, so kind of a retrospective definition. Stability of resistance is already alluded to. Does the type of resistance you have, is it effective over different environments, over different temperature range? Will it be effective under climate change? The usability of resistance, hey, if you're in a national park, you may be able to say, hey, if you can give me 40% survival, I'll take that. I'll plant 3 seedlings to get 1 for my target. If you're a commercial timber company, you might say, well, hey, 25 or 40% survival, that's not going to do it for me. Come back when you have higher resistance. So it really depends on the land manager, what the vision is and what you want in terms of usability of resistance. If we told you there was 20% resistance in a particular species as opposed to none, some people would use that to get the species out there with the hope that it would evolve over time. The trees as they grew up would intermate, the level of resistance would increase. Here's an example. What level of resistance is out there in natural populations? And looking at much of the early work here at Dorena for 2 other species, western white pine and sugar pine, probably in sugar pine, we were lucky to get 5% survival in many of our rust inoculation tests of seedlings. Okay. Western white pine, maybe we got 10% survival on average and everything like that too. So from wild wind pollinated or open pollinated seedling progeny off of parent trees from the wild, the resistance level is pretty low. And so we've gone into breeding programs for both western white pine and sugar pine to increase the level and frequency of resistance. At Dorena, as I mentioned, we've screened or we've worked with all 9 U.S. species, and this is just showing some of the other species and some of the partners, some of the cooperators. What happens, people send us seed, we grow the seed oftentimes for 2 years, we put it in different types of tests sometimes, and then we summarize the data and send it to our cooperators and share that information and give them information based on our experience here. White bark pine, on the right, you see a seedling with probably dozens, if not 50 or so cankers on it. It still has needle spots on it, so very, very susceptible seedling. The photo on the left, I used to tell our statistician, Doug's passed away now, but Doug was a great guy, and used to kid him and say, hey, Doug, I don't need to tell me. These trees, we plant 12 families or offspring of 12 parent trees in a box, and we plant them in 10-tree row plots. So that first box you see there has 12 rows in it. It has 12 families of white bark pine. And you can see there's several rows where every seedling is red and dead. And there's other rows where there's, 4, 5, 6, 7 seedlings that are still green and alive. And so that's the type of resistance that we see in white bark pine. It blows away what we ever saw for the most part in western white pine and sugar pine, even though we screened, over 4,000 parent tree progeny of western white pine and sugar pine, and we've only screened now maybe 1,500 of white bark pine. We're seeing a much higher level of resistance. This is great news for white bark pine and restoration, at least in the Pacific Northwest region of the range, which is Oregon and Washington, where most of this data is from. If you say, okay, Rich, tell me about the geographic trends in resistance. If you look at this map, look at the pie charts. And when you look at the pie charts, look at the green ones with mostly green and blue in them, as opposed to pink, red, and orange. Okay, up in the Olympic area, National Forest area, you have mostly high susceptible white bark pine, at least what we've tested so far. Whereas in the Cascades, you have a lot of the green and blue in the pie charts, a lot of relatively high resistance. So, if you want to know what to plant and how many to plant, it kind of depends where we've been screening, what the patterns are for. It's not going to be the same over the whole geographic range of the species. And so it's good information to have so that each part of the region, each forest, each tribe, each national park, and other entities can plan their restoration. We'll ask some questions about durability and stability. We'll ask how the white bark pine resistance compares with some of the Eurasian pines, like Pinus iberica. You see some of that in a minute. We'll ask, hey, does it matter? You guys put 3,000 spores per square centimeter on these seedlings. What if you put fewer or you put more? Would you get the same answer? And you'll see some slides that kind of address that here in a minute. What's the best white bark pine family we've ever tested? Well, this is one particular trial, and what it shows is 1 year after inoculation, what percentage of the seedlings have cankers or stem symptoms? And there were 120 families in this test, and for the most part, I think the average percent stem symptoms, it will increase after 1 year somewhat, but at 1 year, there were only about 15% of the seedlings that did not have stem symptoms, averaged over all the families. Okay, so really hit this trial hard. There's some photos of it there. But what you can see on a family basis, each of the points in this graph are on a family basis. And one thing you will notice, there's a lot of these families with over 90% infection, stem symptoms or cankering. There are some around 40 to 70%. So those generally are the ones we consider high resistant in white bark pine. Those would blow away almost anything we saw in sugar pine or western white pine. And if you notice at the bottom where that red arrow is, there's one seedling family from the Deschutes National Forest that had 96.7% of its seedlings canker-free or stem symptom-free 1 year after inoculation. It's in first place, okay? Second place is far, far away. So, the question as we get into molecular markers and looking at things further, as Dave was talking about, is, hey, is that due to maybe a single gene? Maybe we do have a single gene in white bark pine for something. Or is it due to, okay, quantitative resistance, but there's quantitative trait loci, and there's one of major effect or something like that too. I always play the skeptic. So I wanted to know, well, is this family really that good? So what we did, we took a few of the boxes with survivors. We put them back in an inoculation chamber several years later. We have re-inoculated them with a higher spore density. And you're going to have to wait for the answer, because in a month or two, we'll be looking at them and see if they have spots and stem symptoms, and we'll follow them next year as well too. So if this is true and they stay mostly this high, that will be very, very impressive for white bark pine. It will be our #1 white bark pine, I think, anywhere in terms of resistance. The other ones that have 40 or 60%, they're very, very good. That's a very impressive level of resistance. But this one from the Deschutes would blow those away. So what happens in some of these species, in this case, Rocky Mountain briscoe pine or limber pine, if you change things a little bit, if you put on a lower inoculum density level, fewer spores, what if you put on the same level, normal level of spores, and what if you put on a high level of spores? What would happen? Well, generally what happens in limber pine, it doesn't make much difference, because most of the resistance in limber pine is due to major gene resistance, and the spore density doesn't really have an effect on major gene resistance. So for limber pine, it doesn't really have an impact, the spore level. You can see on the left side, Pinus aristata, Rocky Mountain bristlecone pine, as you go from a low density spore inoculation, which is green, to a medium density to a red density, you get higher infection and higher mortality levels. So, you know, it's probably quantitative resistance, and it can be eroded to a certain extent, maybe on some of your highest hazard sites. So, you know, each species is somewhat different. The types of resistance are somewhat different, and one does have to be careful making an early generalization based on one test with one level of inoculum density. And that's why at Dorena, we survey these things several ways. Does the geographic source of blister rust matter? Okay, the diagram on the graph on the left shows a range of seedling families and in a Shoshone bulk seed lot that were tested against two very different geographic sources of rust. And as you can see, for the most part, the ones that are resistant with one source of rust are also resistant with the other source of rust as well too. So that is good news. On the right, we had two different inoculum densities, and we asked the same thing about white bark pine. Does it matter if we go up in inoculation density? And as you can see there, for the most part, the resistant families that were resistant under low inoculum density, lower number of spores applied to the seedlings, are the ones that are most resistant under the higher one. What does not happen, there aren't some that were low survival, and when we put lower number of spores on them, they suddenly became higher survivors. And white bark pine, in a sense, is both among the most susceptible species, but it's also one of the more resistant species. The families in white bark pine that are susceptible are very susceptible. You can almost say the word blister rust and that, you know, they'll get infected. But the resistant families can tolerate a lot of blister rust. You'll see another example in a minute. Components of blister rust infection. If we look at several components, traits that we're scoring, and we look at them under low or high spore density, if we look at percent survival, percent cankering or stem infections, number of stem infections, and the percent that remain alive even if they have stem infections. That's what these four graphs show. And they show it at the low and the high inoculum density. And what we'll notice is the Mount Hood and the Colville National Forests seem to do very well in all those graphs, and the inoculum density level does not influence much. Whereas the Deschutes National Forest, the families in this particular trial, the Fremont National Forest, you know, there's almost no resistance in that as well too. So depending on where source you're using geographically, and maybe depending on the range of spore density or number of spores you're putting on your test, you can have different results. But the good news in white bark pine is the resistant families, for the most part, remain resistant regardless of the geographic source of rust or the level of spore density you put on them. This is another way of looking at resistance, looking at it over time, the Colville families versus the Deschutes families. And the population from the Deschutes in this particular trial they all went to 100% or close to 100% stem sometimes very, very quickly. You can see in the Colville population, they only go up to about 50% by the time the diffuse is already close to 100%. And then over time, there is some, this is the nature of partial resistance sometimes, there is some increase year, 2 years, 3 years later in the Colville in terms of infection showing up. So the fungus is being slowed, countermeasures being taken within the seedling or sapling, but it never gets to the level of infection that some of these other populations get to. This was a trial. Again, being a skeptic, I wanted to see what would happen with our resistant versus our susceptible family. So we took 3 resistant, 3 susceptible families. This photo on the left shows 1 resistant, 1 susceptible families. We said, normally we put 3,000 spores per square centimeters on. What if we went down to like 500 or 590? And what if we went up to 24,500 spores? What would happen? You know, with the susceptible at the low density have a lot of survival. Would the resistant ones wilt and get cankered at the real high level? Well, what you can see on the photo on the left is there's 6 rows of the resistant family, and to the right of the yellow line, 6 rows of the susceptible family. And again, you don't need statistics to say, hey, the family on the left is doing a lot better than the family on the right. The graph on the bottom shows 3 resistant families on the left, 3 susceptible families on the right. The susceptible families are much more susceptible than resistant family, regardless of your inoculum density. I think, however, within the resistant families, there are some resistant families that may be more robust under a higher inoculum density. Maybe they have more genes in quantitative resistance, or a different combination of genes and quantitative resistance than some of the other ones. So even there's some nuances going on. You know, not all resistant families are the same. If you were to rank the families, and that's kind of on the far right-hand corner of this table, with 1 being most resistant, what species might be the most resistant that we've looked at here at Dorena? And this kind of agrees somewhat with what Ray Hoff and Richard Stelfon did many years ago in smaller trials. But, you know, Pinus aristata, Rocky Mountain bristlecone pine, and Pinus albicaulis, whitebark pine, are probably some of the 2 species with among the most resistant families. I think foxtail pine, even though it's related to Pinus aristata, maybe are most susceptible of all time. We have a couple large tests, one with 150 families in it, and I'm hoping there will be a couple, you know, I'm hoping there will be 5% survival in that trial, but I'm not sure. We have a lot of dead seedlings out there. The first trial of that species, we had 700 seedlings or something, and only 2 seedlings survived. They both were cankered, but you know, like the top died on one, and I think another one, a branch shot up and whatever too. So I wouldn't have called the 2 survivors resistant. So species can vary in the type of resistance they have. They can vary in the level and frequency of that resistance, and then geographically, the pattern can vary by species as well. Field trials are essential for testing resistance. Some people just do seedling artificial inoculation studies, and depending on your system, whether it's blisterous or elsewhere, you can get some anomalous results. So we've established, as you see, a network of trials throughout Oregon and Washington, and with partners even elsewhere. So that helps you to validate your seedling inoculation trials. It helps you to examine the durability and stability of resistance. It provides a demonstration for field trips of the efficacy of resistance, and it helps you to monitor genetic variation and the impacts of climate change. On the right, you'll see I was at one of our 40-year-old sugar pine trials a month or so ago, and it's a neat series of 6 sites and stuff like that too, and it will really help answer what is the future of sugar pine under climate change and blister rust. The first 40 years, you know, and these things were planted in 82 or 83. So hey, kind of at the, you know, beginning of what a lot of people kind of consider maybe the start of climate change. So hey, how's the species doing? And then these trials, which you'll see in a minute, have a lot of blister rust in them. The photo at the bottom shows a tree with a large vertical canker. It's 40 years old. It's had that canker probably for well over 20 years, but it's living with a canker. So I talked about the importance of field trials. I don't think there's any bluewood of non-native invasive species that has more field trials out than we do. And that's due to a lot of cooperators and a lot of help putting these out and assessing these too. And these maps show some of the locations, some of our trials. We don't have them all on maps like this yet too, but it would just show the network of trials that we have out there. Some of them only a few years old. Some of them, as I mentioned, 40 years old. What happens over time with blister rust? This is part of that 40-year trial series. This graphic only shows them through 25, but there were 6 sites, and I'm showing 3 sites here. One of the sites shown here was rated low hazard before it was planted. One was rated medium hazard. That's the one in the middle, going down vertically. And the one on the far right was rated high hazard before these were planted. And you can see at age, and then there's a time period, age 5, 10, 15, and 25 that these were assessed. And if you look at the age 5, the green squares are individual trees that have no blister rust infection. The yellow squares are trees that have blister rust infection but are living. The red squares are trees that have blister rust infection are dead from blister rust, and blue squares are dead from other causes. But if you look at that top 5-year thing, on the far right, the high hazard site was a winner in terms of the site with the most blister rust. A lot of trees with blister rust infection, some already with mortality. If you go down to age 10, you see things starting to change a little bit. The high hazard site is very impressive. A lot of mortality at that point from blister rust. But the medium high hazard site has crept up in terms of its level of blister rust, both in terms of infection and mortality. On the far right, the low hazard site is doing okay, but it's got some blister rust in there. Everything changes at age 15. At age 15, suddenly even the low hazard site has close to 90% of the trees with infection. Mortality is starting to take off. If you go down to age 25, you can see high level mortality from blister rust, even at the low hazard site. So when you talk about, a low hazard site, high hazard site, as many pathologists that work with blister rust will tell you, there's some art to go with the science in terms of predicting that, and different things come into play there. If you ask, hey, what species are most resistant and susceptible? This is we have some older trials of white pine and sugar pine, and in a couple of the trials we have both species. This happens to be one trial series at 2 sites where we have the same families of sugar pine and white pine at the 2 sites. The red lines are sugar pine, the white pines, or the green lines are western white pine families, and you can see for the most part, western white pine has a lot more resistance than sugar pine. So that's, but it does vary. The level of resistance varies by which site these are at. So again, very illustrative of the differences between families, the variation within families for resistance. On this graph, this is blister rust infection out there. You might say, well, great, you have a great susceptible control. They're all dying there. And what this is the trees that are red are the trees with major gene resistance. This is a trial about 30 miles from a Dorena of western white pine, and there's a virulent strain of rust. And so the trees with major gene resistance have, I think, 100% infection or very close to it. They actually have more infection, or at least at this point in time, than our susceptible control. And then all the trees that are green there, for the most part, are trees with partial resistance. So you can see why you want to look hard at partial resistance or quantitative resistance. And like I say, on some sites, major gene resistance might be okay, and we do have some sites where we're following it and it's still holding up, but you have to be careful if you only have major gene resistance, or you want to promote that big time, you could end up in a situation like we have here. What happens if you take white bark pine, a high elevation species, you plant it at about 1,100 foot elevation, you go through some of the worst drought years in Western Oregon history, you go through some of the hottest years in Western Oregon history, and at least at this low elevation site, 2 different plantings, one a 2011 planting, one a 2015 planting, the species is doing okay. So maybe we'll learn a little bit more about the species, seeing how it does at these sites that have kind of extreme temperatures that you don't even face at the high elevations maybe. And our biggest problem at this site, are gophers. You know, gophers will take out trees that are even a meter tall for white bark pine. And most of the mortality is from gophers and some other vegetative competition. As I mentioned, for success in one of these breeding programs, you have to have a lot of partners, and Washington DNR and some people working with them and us have put in an array of trials up in Washington. I think 3 sites for white bark pine, 12 sites for western white pine. So part of this very, very extensive network I'm talking about. So a lot of kudos to all those partners out there. And some of these white pine trials were planted in 2006 and 7. Some of the white bark pine ones as recently as 2017. They just had their 5-year assessment last late summer and early fall. So again, success. We've had people from universities work with us, from nonprofits, individuals, maybe private companies, et cetera. It all comes, tribes, and a lot of people involved in getting to the successful point of getting white bark pine or any of these species out there in a resistance program. And this is a poster. It's written up in a proceedings as well too. So if you want to see who the cooperators are, but really the cooperators are key to our success. So breeding for resistance, you got to have an assay to screen resistant phenotypes fairly quickly. Eventually, maybe we'll find some molecular markers. I think that's the hope from Dave, Neil, and myself, but for most part, we're not there yet. You do want to validate any of your seedling screening over the long term, so field trials are great for that. You do want to incorporate the full range of resistance. That's why we inoculate young 2-year-old seedlings. We follow them 5 years after inoculation to pull out or separate the different types of resistance, because we want to make sure we're incorporating as many types of resistance as we can, so that we'll have the full array of things to hopefully give us survival out there in the wild. We want to combine, if we can, major gene resistance and quantitative resistance. You can monitor your resistant parent trees in the field. Kind of through this, we found out for Alberta limbered pine, they had sent us some seed to test. They gave us information on what the parent was doing in the field. We tested it. We said, hey, you got a major gene resistant family. I looked back at the summary of the parent tree information they sent us. A couple of those parent trees had cankers on them. So I asked for clarification. They checked those. Yeah, they have cankers on them. Some of them are spore related. So we think, and working with our colleagues up in Canada, including Junju and Lou, with molecular work, we think there's a virulent strain of rust that has evolved for major gene resistance in limber pine that is now present to some extent in some populations, at least in Alberta. So that's where the resistant parent trees can serve as sentinels and be important. So again, we're in the business of developing resistant populations. How can we speed this up? To a certain extent, it comes down to public support, funding, and then the facilities to carry this out. Protecting the parent trees in the field can be very important. For white bark pine, you can go back to these. I think people are putting in seed orchards now, but I think for many populations, maybe not all, for many populations of white bark pine, if you go out and make seed collections over the next 3 bumper crops of seed, you may have enough seed to do most of your restoration work well before seed orchards of white bark pine are in big production, which may take 15, 20, 25 years in white bark pine. Because as you saw in the photos I sent, even at low elevation site, white bark pine grows fairly slow, and it's not going to have a lot of cones on small trees. Acquiring seed, let's build up the seed inventory as soon as possible. For a species like white bark pine, you're going to have to go out to the field for the first 15, 20 years. For white pine and sugar pine, we have seed orchards, and many cooperators have seed orchards. You can, in some areas for white bark pine, you encourage natural regeneration, make sure subalpine fir and other species aren't taking over all the sites, and areas like Mount Rainier, or maybe we'll have enough resistant seeds come in with the nutcrackers that, natural regeneration might be a big part of their management to encourage white bark pine in the future. And then you got to be strategic about where you do your restoration plantings, because you can't replant or restore every acre that's been affected. And so choosing wisely both the seed you use as well as where and how you go about it is really important. Genetic resistant white pine blister rust would be a key on many success to being successful. Each species will have a different level and frequency of resistance. Restoration and genetic trials for a number of our species are underway, some of them up to 40 years old, as I mentioned. Plantings can serve to monitor the potential impact of climate change. One of the key things, we have a number of plantings. There are general restoration plantings that have gone on here for whitebark pine and other species in the Pacific Northwest, but there's a number that we've been involved in that we actually have every seedling tag, so we know the parent tree it came from, the type of resistance it should have, and these can be followed over time. So we can answer potentially the question, hey, what happens over time to that type of resistance, to that genetic background that that family has? What happens under climate change? Do some families do better than others? So a lot of information can be gathered and shared with cooperators and partners if you have at least a subset of your plantings with materials identified. So I think there's a lot of cautious optimism for the future for many of our 5-needle pines in the presence of white pine blister rust. Western white pine, sugar pine, whitebark pine are ahead of the game because the most material have been looked at, screened, and either bred for further resistance, or with whitebark pine we have high levels and we can go back to the field in some areas. But remember, this will vary regionally. And we want to study and understand the long-term impacts of climate change, both on the species itself, but also on efficacy of resistance under climate change. So how do you succeed in these programs? You better find some people who are passionate about the species. When I was giving a talk on ohia and rapid ohia death in Hawaii, that's what I said. That's the key. Public support, you have to enlist public support. A sense of urgency. Okay, that's why I mentioned don't let this get caught up in research. You know, researchers are kind of asking the question, why does something happen? When I started a COA program with some people at HARC in Hawaii and back in 2003, soon afterwards, I was talking to one of the colleagues there and they said, hey, I think we've solved the problem. Okay, I think we have resistance for the most part. However, I can't tell you how many genes. I can't even tell you the underlying mechanism. But, you know, we're cautiously optimistic we solved the problem. If we waited until we had all those underlying answers, they still wouldn't have restoration going on. Now in Hawaii, there's a firm that has built a mill over there, bought some land for they produce high-end guitars and wood for high-end guitars. And so they're investing in koa in Hawaii based on what we have. We still can't tell them how many genes are involved. We still can't tell them the underlying mechanisms. That can now come from the material we produce that is resistant. Somebody can actually look at that and determine what that is. And that'd be great to know. And it may give us some key about whether this is going to be durable. But you don't have to wait for all the answers. Breeding is simple. Hey, I can do it. You can review other successful programs and borrow from those. Proper structure and staffing of a program is important. Cooperators and partners are important. Continuity of funding and personnel. You can't turn this on and off every 3 or 4 years. You know, having a series of 5-year plans and reviews is a good thing. Are we making advances? How can we speed up those advances? And then, of course, you want to be using the resistance seed and restoration. So making sure you're communicating with the land managers out there. And pretty much lastly, here's a planting from 2009 at Crater Lake National Park. And this planting is also a genetic trial. It's a restoration planting. They put it out the way they wanted. We've got every seedling tag. There's a little blister rust in there. Gophers have found some trees. Nutcrackers are scoping the area out. This planting is right at Rim Village. So if you go to Crater Lake, you will see this planting. And it's also ADA accessible. There's walkways in between it and everything like that. So for public conservation education, potentially up to 400,000 people see this planting a year. Eventually they'll know, hey, does resistance work? What is the future of whitebark pine? So, you know, envision the future. We have an opportunity. We can do nothing or we can do something. All of you that are helping with resistance program, all the way from the start of helping select trees, to those doing testing, to those putting field studies out, to those actually doing restoration work, you're all chose to do something to help the species. Genetics is the key. Let restoration begin. This is a list of some references, literature. There's a lot more that I couldn't fit on here too, so it's not meant to be a comprehensive list. The first paper on there is kind of a overview that this talk is based on, so it's a good starting point. acknowledgements, like I say, a lot of people to thank and whatever and certainly the Forest Service for both the Forest Health Protection Program as well as the Genetic Resource Program. Very, very instrumental in terms of keeping these programs going, in terms of the success that we're seeing to date. So thank you.
All right. Thank you very much, Dr. Sniezko, for that wonderful talk. Unfortunately, we are almost at time, so I think we have time for one question, and I see a raised hand, so I will allow Rick to ask. Go ahead, Rick. Can't hear you. Alrighty, let's ask, or I'll read a question out from the chat. Jody says, thanks for sharing so many amazing achievements and great ideas, Rich. Might have missed it. In Sniezko et al. 2008 graphs of natural resistance levels across families of 2 species. Were those samples totally unselected, or were those selected plus trees submitted for testing?
They were the 2008, the Western White Pine, Sugar Pine. They were wild selections that were made probably in the 60s and 70s. Each area in Oregon and Washington had a different level of blister. So in theory, they were plus trees that were selected in different areas, different levels of blister. So yeah, they're probably better than just a random selection, but in some cases, maybe not a whole lot better.
Gotcha, thank you. And then Rick asked, on the Inyo National Forest, as far as I know, there's no white pine blister rust. Has there been any work on the trees there?
You're talking about internationally. There, over in Eurasia, there is blister rust. In China, there's blister rust and some blister.
Sorry, on the Inyo National Forest.
Oh, which one?
Inyo.
Okay.
Sierra Nevada.
Okay, and what was the question about that?
Has there been any blister rust related work there?
You would have to ask colleagues in California. I suspect maybe the sugar pine program is the most advanced in California, and they've done a lot of testing for major gene resistance in sugar pine, and they've collected throughout the state, there, including Southern California as well too. Western white pine, they've started more recently looking for major gene resistance. There's a little bit of work going on in some forests for white bark pine. The national parks are starting a project with white bark pine. So for Inyo National Forest in particular, I'm not real familiar with, but John Gleason at the Placerville Nursery has done the screening work for the California species for the most part.
Awesome. Thank you very much. Unfortunately, that's all we have time for today. Please join us next time on March 21st when we will hear from Shinasha Pete from the Confederated Salish and Kootenai tribes about sowing history, growing culture, and planting language, CSKT's white bark pine biocultural restoration. Thanks again, everyone, for joining us, and we will see you next time.